Welcome back to part 3 of chapter 3. In this section, we're going to be focusing on aerobic respiration, which is the main mechanism our body uses to produce energy. But we will also look at anaerobic fermentation at the very end of this chapter. So aerobic respiration or cellular respiration is a pathway, a metabolic pathway that has multiple steps. So I think I've talked to you about metabolic pathways and how they are very branched. The pathway is not from A to B, from B to C, and from C to D, and we're done. It can go from A to B, or A to C, or A to D, or A to Z, and then from there, depending on which route you're taking, you may then go to X or F or L. It just depends. So metabolic pathways are very branched and they're very complex. And this is one pathway that is used to produce energy. It's the most common pathway we use to produce energy. And we use glucose as our organic molecule. Glucose gets oxidized completely, and ATP is produced in the process. This is an aerobic reaction, which means we need oxygen. Aerobic means needing oxygen. And so we are going to oxidize glucose, and we're going to reduce oxygen. So remember, oxidation and reduction actions always go hand in hand. One thing is oxidized, something else is going to be reduced. We don't oxidize glucose in one step. It takes multiple steps to oxidize glucose. But the end product will be carbon dioxide, which is completely oxidized. So here's your glucose. That's incorrect. Hold on one second. That looks much better. So C6H12O6 is glucose, and then we have 6 oxygen molecules. And we're going to oxidize glucose to produce carbon dioxide. So we still have, wow, that was loud, wasn't it? Hold on while I check my laundry because that's what that was telling me. So glucose is going to get oxidized completely. We're going to get rid of all of those hydrogens and the electrons, and we're going to produce carbon dioxide. Oxygen is going to be reduced completely. We're going to add those hydrogens and electrons from the glucose to the oxygen, producing water. This is a redox reaction. So there are 2 basic ways that we're producing energy in cell respiration. One way that produces a small amount of energy is called substrate level phosphorylation. And so substrate level phosphorylation is just what it says. We take a substrate and we add a phosphate to it. The substrate is ADP, adenosine diphosphate. We add a phosphate to it, producing ATP, adenosine triphosphate, which is the energy currency our cells like. So this is an anaerobic process, and it happens during cell respiration in a couple of the basic stages. The more common method of producing energy in aerobic respiration is through oxidative phosphorylation, where we transfer electrons from one carrier to another carrier, and in the end we use oxygen as our final electron acceptor, and in this process we produce a lot of energy in the form of ATP. So there are 2 regions that aerobic respiration occurs in. One is in the cytosol. The cytosol is the fluid-filled region between the plasma membrane and or within the plasma membrane, that's what I'll say. It includes all of the organelles that sit inside of it. So in the cytosol, we have one phase called glycolysis that occurs. And then the other three phases of aerobic respiration occur within the mitochondria. So the mitochondria is this kind of kidney bean looking structure. It has a double membrane, an inner and an outer membrane. And the intermediate phase or transition, citric acid cycle or Krebs cycle, and the electron transport chain all occur within the mitochondria in specific regions. So all of these mechanisms, or all of these phases, I should say, need oxygen in the mechanism. Whereas glycolysis here does not need oxygen. It's an anaerobic process, so without oxygen, not oxygen requiring process. So let's talk about these phases. Glycolysis is the first. It occurs in the cytosol of the cell. It does not require oxygen. And in this process, glucose is broken down to 2 3-carbon molecule pyruvates. Pyruvate then is going to either move into the mitochondria, or if there's no oxygen available, it's going to be reduced further. So this pathway contains 10 different enzymes, which means there are multiple reactions. This one pathway is very complex. Anytime we want to make energy, we have to first give a little bit of energy. So think about this morning when you got up and you were like, oh my goodness, I am so tired. I need energy. I need to do something. And what did you do? Think about that. Just kind of ponder over that. So I know what I did, and I'm going to share that with you in a second. I chose to boil some eggs, and then I used salt and pepper to flavor the eggs, and I ate them. So I did boiled eggs. That was super yum, and I made some coffee with some creamer, and I was like, yay, right? Because it was really good. I used energy, though, to gain energy. I had to use mechanical energy to actually get the food cooked, and then I used more mechanical energy in the form of chewing the egg, swallowing the egg. My cells had to break down the large macromolecules, protein, as well as the fats in the cholesterol portion, the yellow. And then I was able to gain energy in the form of ATP from my food that I ate. So I had to invest energy to gain energy. In this process, we invest 2 ATP, which activates the glucose. Glucose is then converted to an activated 1,6 glucose phosphate biphosphate molecule, which then immediately breaks down into 2 glyceraldehyde 3-phosphate molecules. Glyceraldehyde 3-phosphate molecules are then going to be oxidized to produce the pyruvate. And if you look here, I told you that C6H12O6 is glucose. Here's C3H5O3, which means we produce 2 of these molecules, so we still have the 6 carbons, we still have the 6 oxygens, but we've lost 2 hydrogens. So glucose was oxidized, which means we had to reduce something. So G3P is oxidized, and in this process we produce some ATP in the form of substrate level phosphorylation. So when we oxidize this molecule, we also take the phosphates that were added and we produce ATP. So we get a total of 4 ATP produced, but we only gain 2. So here's the initial substrate. The end product is this. We have 2 hydrogens that we are not accounting for. We gained 2 ATP because we invested 2. We formed 4, but we had to give back 2. And anytime we oxidize one molecule, we have to reduce something else. So NADH is formed from NAD. NAD is one of our coenzyme carriers that picks up electrons and hydrogens when we oxidize a molecule. So it's kind of like a taxi. Picks them up, and its job is to go into the mitochondria to the electron transport chain, drop off its hydrogens and electrons, and then go back to where it came from. So here is the inputs and outputs for the glycolytic pathway. Do you see this? I have a song here. I'm a little scared to play all my songs because if I play them, the initial artist may say, oh, I don't want you playing that on your video. So I'm not going to play it now, but it's a good song and you should listen to it. I may play it in class. I know, I'm not sure. So glycolysis, I'm saying so a lot, I notice. So as I say so, I actually use the word so to explain that I'm saying so a lot. Bizarre. Regulation of glycolysis is controlled through allosteric inhibition. If we have enough ATP, we don't need more ATP. If we have enough NADH, we don't need more NADH. Then we're going to shut down the glycolytic pathway. One of the enzymes in the glycolytic pathway is called phosphofructinase. And we turn that one enzyme off and the entire pathway ceases. It doesn't work until we need it to work again. If we need more ATP or more NADH, then we're going to remove that glycolytic inhibitor and phosphofructinase will be able to work again and we'll be able to run glycolysis. I told you that at the end of glycolysis, pyruvate is either going to move into the mitochondria, or if there's not enough oxygen available, it will be reduced. So there are 2 fates of pyruvate. If you look on your study guide, one of the questions I have on there is, know the 2 fates of pyruvate. So part of the response is right here. The fates of pyruvate depend on the availability of oxygen. Without oxygen, pyruvate does not enter the mitochondria and instead is going to be converted to lactate. This is to allow us to produce, or I should say reproduce, NAD. So NAD was reduced to NADH, but if there's no oxygen available, NADH has to be oxidized back to NAD so we can produce oxygen through the breakdown of another glucose molecule. If we have oxygen available, then pyruvate enters the mitochondria and we'll go through the next 3 steps. I'm sorry. And we will produce more ATP through aerobic respiration. I'll talk about this a little bit later again. Here's the glycolytic pathway ending with lactic acid production and NAD production as well. So the next step, or the next step, is within the mitochondria. We're going to move into the mitochondria and into the region between, or not between, inside both of the membranes. So we have this outer membrane and this inner membrane. We're going to go inside of the inner membrane to the matrix. This is where the preparatory or transition stage occurs, as well as where the citric acid cycle occurs. The preparatory reaction is a reaction that takes the pyruvate and converts it into a 2-carbon molecule. That's the job. The only type of molecules that can enter the citric acid cycle are 2-carbon acetyl groups. So the 3-carbon molecule, pyruvate, is converted to a 2-carbon acetyl group. And that's going to be carried by another coenzyme called coenzyme A. So pyruvate and coenzyme A, they interact, they mix together, they kind of do a little dance. And in the process, carbons are removed. So we have decarboxylation occurring. We also have oxidation of the pyruvate, forming the acetyl group that attaches to coenzyme A. Anytime we have carbons removed, we're going to need oxygen available. I just mentioned this. I'm going to get to the next slide. Here we go. So here we have pyruvate and coenzyme A. These two mix together. In the process, we remove hydrogens. So that's oxidation. NAD is reduced. Pyruvate is oxidized to the acetyl group. NAD is reduced to the NADH. The carbons that were in the 3-carbon pyruvate that are removed are going to attach to oxygen, producing carbon dioxide. So now we have 2 acetyl groups that are attached to coenzyme A, and they are going to enter the citric acid cycle. So the citric acid cycle is a cyclic pathway, also known as the Krebs cycle for the individual that found it. This is a cyclic pathway that starts with a 5-carbon molecule. Oh, I'm sorry, I said that wrong. A 4-carbon molecule called oxaloacetate. We have a 2-carbon acetyl group that is released into the citric acid cycle by coenzyme A. Coenzyme A goes back to the transition phase where it can then pick up another acetyl group. Oxaloacetate and the acetyl group connect, forming citric acid, which is where we get the name. We have this citric acid molecule that then is completely oxidized back to the oxaloacetate. All the carbons that were added from the acetyl group are completely removed. So the only molecule we have left at the end is that oxaloacetate, but we have a lot of these coenzymes. We have some waste product called carbon dioxide, and we produce ATP. Here you have oxaloacetate, acetyl-CoA, they mix together, forming citric acid. Coenzyme A leaves, and we immediately remove one of the carbon dioxides, or one of the carbons, give it to oxygen. NAD is reduced, so we remove hydrogens, remove the other carbon, remove another hydrogen, produce some ATP, remove another hydrogen, and the thing we have left at the end is this oxaloacetate. This cycle occurs twice because each pyruvate that came in to the transition phase produces one acetyl group that will then be oxidized completely, and the only thing we'll have left is that oxaloacetate. This occurs in the matrix of the mitochondria. It requires oxygen. Acetyl coenzyme A is the initial substrate. Carbon dioxide, coenzyme A are produced. ATP, NADH, and FADH are formed through the oxidation of the acetyl group. Oxaloacetate is going to be involved in the process, and it is going to be regenerated in the process. So for every 2 turns of the citric acid cycle, 1 glucose molecule is going to release 6 NADH, 2 FADH, and 2 ATP. Yeah. I don't have a video here. I'm not sure why I have a star on that one. Sorry. Citric acid cycle is also regulated. We regulate it using citrate synthetase. If we have high levels of NADH or ATP already, then we're not going to need this pathway to run. But if we don't have high levels of NADH or ATP, if we need more, then the cycle is going to increase. At this point, we have completely oxidized glucose. It's been broken down completely. There's nothing left of the glucose molecule. The carbons that were there are released to oxygen, producing 6 carbon dioxides. The oxygen that was there is going to be used in the reaction to oxidize the NADH. And we're going to move into the electron transport chain. I'm going to stop here because this is almost 22 minutes now. So I want to stop and we'll get the last bit in section 4 or part 4 of this video, okay?